And the next step for this is to integrate into the genome. This is all made in the cytoplasm, this nice double-stranded DNA. And then, and that's shown on the top here, with the two LTRs, the viral DNA is in blue. And this is going to integrate into the host cell DNA. So the target here is our DNA or any animal's DNA. And the integration target is this yellow, this orange bit right here in the middle. The rest of the genome of the host cell is in purple. Once this integrates, and we'll look at how this happens in a moment, we now have an integrated or proviral DNA. Remember, the provirus or proviral DNA is the DNA integrated into the host cell. So now you see we have host DNA on either side. Remember this orange part that was the target? This is now duplicated on either side of the provirus. We have one orange sequence here and another one here. The duplication is a feature of the integration process, which I'll tell you about in a moment. And when you see duplicated sequences like this, the first thing you should think of is it's a retroviral integration. And in fact, that's how we tell if sequences in our genomes are retroviral or not, by looking for duplicated host sequences on either side. We also have the two LTRs, which are shown here in green. And then we have the rest of the viral DNA in the middle, proviral DNA. Now the LTRs are important because in building them into the DNA, what we have done is to build a promoter at the 5' end for RNA polymerase II and a termination and polyadenylation sequence at the 3' end. So now in this integrated state, host RNA polymerase will come along and say, aha, there's a promoter here, and all those sequences that regulate transcription, they're all there, and transcription proteins will bind, and the host pol2 will come here and start making mRNAs, pre-mRNAs. They're capped, they cover the entire genome, and they're polyadenylated. So again, this is all done by the host cell, by the host cell RNA polymerase. And it wouldn't happen if we hadn't generated these LTRs by that Baroque reverse transcription process. So let's look at the integration. Now this is carried out by another viral enzyme called the integrase, which is a separate protein. So far we have reverse transcriptase and RNase H, one protein. Now we have integrase, it's a separate one. And what this does is take the DNA, the double-stranded DNA that we have made by reverse transcription, and integrates it into the host cell DNA. So here's an outline of the integrase protein in black here. And here's retroviral DNA. So the retroviral DNA is shown with its ends adjacent. The integrase catalyzes the invasion of the host DNA, which is labeled here as target DNA. It's purple. That's our genome or whatever genome of a cell that's going to be integrated by this viral DNA. It catalyzes an attack by the 3' ends of the viral DNA to this double-stranded target DNA. And that breaks the target DNA. It generates two free 5' ends from the target DNA. You can see on the upper right. They are then ligated to the viral DNA by the integrase protein. It is then repaired. So now we have two gaps here, right, between the viral DNA and the host cell DNA. Those are then repaired by the addition of nucleotides and ligated. So now we have a provirus. So you can see by virtue of the fact that we are invading and nicking one strand of the host DNA duplex and then filling it in, we're duplicating the sequence at either end. That's where the duplication comes from, because you're copying each strand separately to repair this. So those orange target sequences are duplicated precisely by this mechanism. Now it also turns out that in order for this to occur, the viral DNA has to be chewed back a little. So you can see here the 5' and 3' ends are flush, but the integrase chews back one strand, the plus strand, a few bases, and you can see that's why you have an overlapping 5' strand here. So that you lose a couple of bases at the viral ends, and then you duplicate the host cell target sequence. So that's all done by the integrase protein. So this is the X-ray crystal structure of the integrase protein. It's a dimer, as shown on the left here. And the way it interacts with the viral and cellular sequences is shown here. So This is sort of, on the right is an outline of the X-ray structure shown here. The two dimers are shown in green and blue. And so here we have the viral DNA ends laying across the catalytic surface of the integrase. So there are two catalytic cores to dimer. Each monomer has one catalytic site. You need two to accommodate both strands. So here's the viral DNA. That's our 3' end. It's chewed back a couple of bases. We have a free 3' hydroxyl that then attacks the double-stranded host DNA, which is laying across the enzyme perpendicular to the viral DNA. And this attack occurs from both viral strands. The host DNA is nicked. It's then ligated to the viral DNA, and it's repaired, and that gives you the duplication at the ends. And so that's shown up here. So this is all done by the integrase. The reverse transcriptase is finished making a double-stranded DNA. The integrase then comes in with the viral particle into the nucleus and does this and integrates this DNA into the host cell DNA. And that becomes the provirus. So both of these enzymes, reverse transcriptase and integrase, are targets for antivirals used to regulate HIV infection, because these are unique enzymes that we don't have in substantial amounts. Now, earlier, a lecture or two ago, someone asked about retroviral integration sites. So here's a table that addresses that. This is a table where we're looking at retroviral integration site preferences. And so this is a analysis of human cells or mouse cells that were infected with a couple of different retroviruses, ASLV, a mouse. This is an avian retrovirus, a mouse retrovirus, and HIV, of course, human retrovirus. And then the integration sites are mapped. And they're mapped to know whether they are within genes or at transcription start sites. And these are expressed as percentages. So for example, if you just threw DNA randomly into cells and allowed it to integrate, these are the numbers you would expect simply by the numbers of coding regions and transcription start sites within our genome. 26% of the time we'd expect to be within a gene, and 5% of the time within a transcription start site. Now you can see the numbers are not the same as randomness for these viruses. So ASLV, for example, within genes is slightly higher than randomness, as are these other as well. So there's a slight preference for being in a gene. Some of these also seem to prefer being at a transcription start site, but some of the, obviously 8 and 10 are not very different from 5. So the bottom line is that integration is more or less random. There are no specific integration sites, as there is for lambda in its host. The at site in the host DNA is the specific integration site for lambda. There's only one place where lambda can go. These retroviruses will go just about anywhere. They seem to prefer to be within genes, and maybe some of them near transcription start site. Now on the right is an experiment done in mouse cells which were infected with HIV. And you can see 62% integrations within genes, 6 within transcription start sites. So it likes to be within genes in mouse cells, and doesn't really care if it's within a transcription start site. And then a mouse cell line was studied which is deficient in a gene encoding this protein LEGF. LEGF happens to be a protein that's part of the nucleosome in chromatin. And so when you take away this protein, you can see the numbers shift a little. The percentage of integration within genes goes down, and the start site integration goes up. The suggestion here is that maybe LEGF is helping to target viral integration to chromatin, which it might want to do. And in general, what we think is that there is some preference for integration into DNAs that are wrapped around a nucleosome and which are exposed, as opposed to the intervening nucleosomal sequences. So LEGF is bound to the nucleosome itself, so maybe this is telling us that HIV likes to integrate there. Once the viral genome is in the cell as a provirus, it's transcribed, as I said. There's a strong promoter in the left LTR, which is built during reverse transcription, as I said, and that is transcribed by host systems. And the viral mRNA then it can be translated, it can be spliced, or it can eventually be incorporated into infectious virus particles. So I like to say there's no DNA replication and there's no RNA replication. All the virus does is to copy its RNA genome into DNA, because really it doesn't have any other enzyme that is amplifying its genome. So that's an overview of this whole process. Again, starting from entry, we have this core, subviral core in the cytoplasm. All of the enzymatic steps that I've shown you for reverse transcription are carried out in the cytoplasm. These double-stranded DNAs then go into the nucleus, and the integrase goes with them, and that, of course, makes the integration site into the genome. And then we have transcription, which produces mRNAs for various proteins, as well as new genomes. So the genomes, which are going to get packaged into new virions, they get packaged as dimers again. These are full-length transcripts. In other words, full-length unspliced transcripts of the provirus. They have a cap and they have a poly A tail. But then once they're in the particle, they're not going to serve as a messenger RNA later on.